Okay, so quickly just finishing up where we left off on the last one, when we got cut off before we could finish talking about monoclonal antibodies. Basically, they all have the same variable regions of their antigen binding site, because they all come from a clone of a single B cell. They're usually used to treat autoimmune diseases and cancers. They're produced by injecting an animal, usually a mouse, with an antigen. The animal develops antigen-specific B cells through their adaptive immune system. These B cells are then fused with a myeloma cell line, which is just a type of malignant plasma cell, to produce antibodies of the same specificity as the fused B cell that came from the mouse, so they can be used in humans. And these monoclonal antibodies are typically of the IgG isotype, and they treat autoimmune diseases or malignancies by opsonizing them, activating complement or initiating antibody-dependent cell-mediated cytotoxicity by natural killer cells. So moving on, we're going to talk about the adaptive immune system's viral immune response. And these are just some of the subheadings of the topics that we're going to talk about for the viral immune response and the different mechanisms of it. So, starting with the interferons, if you recall from one of the first few videos, we talked about the role of pathogen recognition receptors on cells, which are proteins that detect molecules produced by pathogens or produced by damaged cells. These pathogen recognition receptors pick up these pathogenic molecules, and they present them to cells of the immune system, and then these cells of the immune system respond by producing interferons or cytokines, or activating other kind of cells, or bringing cells to the site of infection. So with regards to interferons, type 1 interferons are a group of cytokines consisting of several alpha and beta interferon molecules or proteins that respond to a stimulus associated with a viral infection. So, for example, if they detect double-stranded RNA, which is indicative of a viral infection, a cell is going to produce type 1 interferons, and then these type 1 interferons are going to initiate an antiviral pathway to try and get rid of the pathogenic virus. So some types of type 1 interferons include interferon alpha, interferon beta, interferon epsilon, interferon kappa, and interferon, I forget what the Greek W symbol is, you'll have to look it up. Once antiviral pathways are activated, the main two type 1 interferons that are produced are interferon alpha and interferon beta, and they're produced by all virally infected cells, and they initiate the antiviral state. This antiviral state inhibits viral protein synthesis, so viral proteins can't be translated to perpetuate the viral infection. So it's a way that infected cells are going to produce these type 1 interferons, alpha and beta. These interferons are going to stimulate an antiviral state within the cell to try and get rid of the virus by inhibiting protein synthesis and activating natural killer cells that will kill the infected cell, or dendritic cells. An impaired or a delayed antiviral response can result in severe illness, and we see this a lot with people who are old, or people dealing with immunosuppressive treatments for diseases like chemotherapy to deal with cancer. Natural killer cells are recruited to the site of infection by chemokines and interferons, like we talked about here. Interferons alpha and beta can attract natural killer cells to the site of infection. Some virally infected cells will downregulate expression of MHC class 1 on their cell surface, and because if natural killer cells don't detect any of this MHC on the cell surface, they're going to induce apoptosis of these cells and kill them through, I believe, perforins or granulozymes to degrade the cell membrane of the target cell that isn't producing any MHC because it's being virally because it's been downregulated by the cell to indicate that it's virally infected, and then the natural killer cell is going to kill the infected cells. Binding of antibodies to virus particles on the surface of infected cells can also trigger a response that we talked about when we talked about the B cells. When we talked about natural killer cells in depth, and that is the antibody-mediated cytotoxicity. And this is just where natural killer cells will bind to the Fc region of the antibody, so the kind of bottom part of the Y shape. They'll bind to it and then they'll release signals or chemokines or granular contents. They're going to trigger apoptosis in the target cell. Moving on to the role of helper T cells in the adaptive immune system's viral immune response. When antigen-presenting cells, like dendritic cells, present viral antigens on MHC class 2 to helper T cells, naive T cells with antigen-specific antigen binding sites on their T cell receptor get activated, typically in the thymus. These activated helper T cells then undergo the process of clonal expansion to produce lots of duplicates to replicate themselves a lot, and these daughter cells of these activated helper T cells are going to differentiate into either effector T cells that secrete cytokines such as interferon gamma, which is going to activate other immune cells like macrophages, or it may kind of stimulate them to differentiate into plasmablasts, which are the short-lived versions of plasma cells, or into memory B cells. Specifically, the interferon gamma is going to activate macrophages and stimulate isotype switching of B cells to produce immunoglobulins capable of opsonizing proteins like immunoglobulin G, which we went into in depth when we talked about isotype switching. And recall from one of our earlier videos that the main helper T cell that's interested in viruses is going to be Th1 helper T cells because they're focused on intracellular pathogens. Virus-specific naive CD8 cytotoxic T cells are activated upon detection of an antigen on MHC class 1 on an antigen-presenting cell. Activated cytotoxic T cells undergo clonal expansion, same as helper T cells, and they differentiate into effector CD8 T cells, which then kill virally infected cells either by releasing granular contents that disrupt target cell membrane integrity like perforin or granzyme, or by delivering death signals to infected cells. Because cytotoxic T cells actually kill antigen-presenting cells, viral infection of slow reproducing nerve cells are going to be a lot more problematic than short-lived and abundant epithelial cells. I was hoping that we'd be able to finish this in one, but I think I'm going to cut the last two parts on B cells and viral evasion in the adaptive immune system into its own shorter video.